you remember that reaction that you talked about earlier in the chapter? How there was, like, there's hydrolysis reactions, or there's dehydration reactions, things like that. So one was called a synthesis reaction. Do you remember that?
That's right here.
Synthesis or dehydration reaction.
Which
So
one?
the synthesis reaction was when you were putting two things together. So the synthesis of proteins are going to be how the protein is formed.
So like the amine group, the amino acid,
So you have amino acids that you're going to put together to make the protein. There's
okay, so that
your amino group and your carboxyl group, which is an acid. Hydrogen, and our R group, which is just what makes this guy different than other guys. So it's just in something else because it varies. But so what happens is we have one carboxyl from one amino acid and an amino group from another one. These guys come together, make water, and then a bond is formed. So this bond that was to this hydrogen is going to bond to that.
that's a peptide bond.
And that is a peptide bond.
Okay.
So this peptide bond is the bond between the amino acids.
Okay.
Then there is the protein folding, which is that page, that picture right there. It shows you, so first it's right after the ribosome made that peptide chain. So we have covalent bonds make the primary structure, so those peptide bonds. Then the secondary structure, which causes the pleated shapes. So if you were to take a piece of paper,
Right.
kind of get it like that, either pleated or helix, is caused by the hydrogen bonds between, so that peptide bonds, hydrogens, and other oxygens, so it kind of makes a shape.
So what does it do? Because both in the pleated sheet and the helix, there's hydrogen attached to the oxygen.
So kind of like those water molecules, how there was those things, those hydrogen bonds. That's the exact same thing that's happening there, is that there's hydrogen sticking off, and there's oxygen sticking off, and then they form hydrogen bonds. Or because of their charges, they're attracted to each other, forming those hydrogen bonds.
So it all depends how it's kind of formed.
Yeah. So it depends on the order of amino groups, because they all have different little things about them.
Right. I think
And
T
so-
goes with A and G goes no. A
So
goes-
you're thinking DNA,
Oh,
okay? So if I make a protein, I can have, how many amino acids are there? There's like 20 something. Isn't there like 21 or something?
no, there's 20 and there's 9 essential and the rest are 20
Okay.
amino acids.
20 amino acids. So different orders of combinations create different proteins. And so based upon the order that they're formed, the hydrogen bonds are going to interact differently, forming a different shape. And the end shape is going to do a different structure, causing different function.
Okay. So it all depends on how they're, I guess, put together, the order of them, that will give it its pleated sheet or its helix.
Correct. And then, so that's the secondary structure. Then the tertiary structure is the interactions between that molecule or that peptide bond, that polypeptide, and its environment. So the different charges and molecules that are around it are going to cause it to bend and make a different shape than what it currently is, causing the tertiary structure. And then, so that tertiary structure can combine with multiple other tertiary structures.
So you can combine like, let's say,
this tertiary structure and this tertiary structure and this tertiary structure come together, and they push and make each other change more shapes.
Into
And so then that gets your quaternary structure, or quaternary.
coronary. Is that because there's only 4?
So that's the fourth
This is the
step.
fourth
So
step.
primary, secondary, tertiary,
Tertiary,
quaternary.
And it'd be just putting those 3 together just to form something.
Yeah.
Okay.
So that's going to be the different steps of its folding.
Okay.